DNA fingerprinting or DNA typing is a technique involving chemically dividing DNA into fragments which form a unique pattern and then matching that identity profile with the pattern obtained from similarly testing a suspect's blood. This technique was developed by Dr. Alec Jeffreys of England in 1985 and is also accurate that a possibility of two pattern matching is 1 in 30 billion. Specimen from living buccal epithelium most common. Blood, hair follicle with a root. From dead, unclotted blood in relatively fresh dead blood bodies, spleen and muscle if unclotted blood is not available. Brain tissue in intermediate postmortem interval, bone marrow from molar, pulp of molar teeth in advanced decomposition. Specimen is collected in a sterile tube and frozen at to minus 20 degrees Celsius if a delay is likely in examination. Preservative used is EDTA as it inhibits activity of nucleases that can break down DNA. Sources of DNA: primary tissues, blood, soft tissue, bone tissue, teeth, bone tissue, bone marrow, soft tissue, spleen, brain, muscle, skeletal tissue, cardiac tissue. Then, principal procedure remark: a restriction fragment length polymorphism. Restriction fragment length polymorphism RFLP method. Blood sample taken. Then isolate DNA. DNA cut into fragments by a restrictive enzyme. DNA fragment separated into bands using gel electrophoresis. DNA band pattern is transferred to a nylon membrane using southern blotting. Radioactive DNA probe. DNA probe bind to specific DNA sequences, that is hybridization. Wash. Excessive DNA probe washed off. Radioactive probe is bound to the DNA pattern on membrane. X-ray film placed to the membrane. Autoradiography of DNA pattern, DNA fingerprint, visual and computer analysis of DNA profile. Most human DNA, 95%, is useless and is considered junk DNA or filter DNA or nonsense DNA. The junk DNA mostly consists of a repeat of short sequences of base in a given length of DNA called as a satellite DNA. This satellite DNA exhibits high degree of variability, polymorphism, in pattern of repeat sequences called as variable number tandem repeat. Number and size of variable number tandem repeats varies from individual to individual and is unique to an individual. Isolation, extraction of DNA, disruption of cells and fractionization of the cellular organelles, dissociation of DNA from a protein by the use of salt solution or detergent, addition of an extract to phase separate the bulk of protein from DNA, use of enzymes of differential precipitation to remove the RNA and polysaccharide. Isolated DNA is quantified by UV spectrophotometry. Cutting and sizing. DNA is cut at a specific sites by a restriction endonuclease enzyme, which recognize specific sequences in a double DNA is cut at specific sites by a restriction endonuclease enzyme, which recognize specific sequences in double-stranded DNA into various fragments called as restriction fragment length polymorphism RFLP, which are produced due to presence of variable number of tandem repeats. Sorting by gel electrophoresis. Cut DNA is on a agarose gel electrophoresis. Different restriction fragments are separated, varying in length between 0.5 to 25 kb, and these fragments may vary from one individual to another. Small fragments move faster than larger one. Agarose gel is then stained with ethylene bromide for 40 minutes, which lightly binds to DNA and fluorescence under UV light. Transfer of DNA to nylon, that is southern blotting. DNA is transferred from agarose gel onto nylon membrane using either capillary transfer technique of southern or a vacuum blotting of transfer, resulting in a mirror image of replica of fragment distribution. DNA is then fixed by heat at 80 degrees Celsius and cross-linked by cation of UV radiation. Advantages of restriction of fragment length polymorphism, very specific test. Disadvantages: require samples in a good condition. Thus, limited value in testing cadaveric tissue for identification of human remains. Isolated fragments are in a ranges of 2 to 20 kbps. Take weeks to perform. For every probing, membrane is stripped off previous probe and re-hybridized, and autoradiography is performed again. Most common method. This hybridization is a pairing of two complementary single-stranded DNA to double-stranded DNA by addition of a radioactive P32 marked probe to the nylon membrane. This probe scan all DNA and hybridizes complementary sequence, make them radioactive. Washing. Loosely bound or excess probes are washed away with 0.05% SDS. Membrane is then wrapped in the saran wrap and placed in the X-ray cassette holder along with an X-ray film kept at 80 degrees Celsius. Exposure time depends upon the specific activity of probe, ranging from few hours to days. X-ray films are then developed and fixed in respective reagent and finally washed in water and dried. DNA fingerprint. Radiograph obtained shows grey to black bands where radioactive probes are hybridized to fragments bearing complementary sequences. Series of bands seen on film is called autorod, which represents DNA fingerprint of that individual and is unique to him. Polymerase chain reaction. Double-stranded DNA to single-stranded DNA, that is denaturation, then cooling, annealing process, primer attached to the DNA, and extension DNA, polymerase extend, primase, heat and repeat. That is method of polymerase chain reaction. Small amount of DNA is amplified a millionfold using thermal Taq polymerase. It does not accomplish DNA typing but increases amount of DNA available for typing. DNA is isolated and split apart by heating sample to more than 94 degrees Celsius. This is followed by a repeated cycle of temperature variation to help DNA synthesis as follows. Temperature is lowered, during which a small fragment of DNA primer bind specifically to DNA. DNA primer acts as a starting point for DNA duplication process. Temperature is raised again to 72 degrees Celsius, causing DNA polymerase to extend the primer and copy two separate strands of DNA. DNA polymerase insert nucleotide opposite appropriate bases, ensuring complementary base pairing. The cycle is repeated 30 times or more, synthesizing more than million copies of DNA by electrophoresis. VNTR polymorphism is done followed by visualization by fluorescent detection or a silver staining. Advantages: sensitive and quicker method, analyzing 36 samples at one time, relative simple and easily extracted outer, permit analysis of extremely tiny amount of DNA. Disadvantages: not specific, however, not much conclusively excludes susceptible to contamination. Most PCR loci have fewer alleles. Some of PCR loci are functional and genes. Application: useful when a very small amount of DNA or partially degraded biological sample is available, particularly used for diagnostic purpose. Then, short tandem repeat. Short tandem repeat method: blood, cell, DNA, PCR mix, denaturate, extension, annealing, adenine, thymine, gynin, repeat, 8 repeat, short and repeat, electrophoresis, lasers and computer system. Then blood, cell, DNA, PCR എടുത്തു, denaturation, then thermal DNA, amplified DNA, electrophoresis, laser system. Okay. Short tandem repeat method. Human genome contains a tandem repeat of DNA sequences which show variability in different individuals. Isolation of DNA, replication of STR fragment by PCR, electrophoresis, identification of STR fragment by staining or laser technique. Advantages is rapid, requires small sample, can also be applied to degraded DNA. DNA fingerprinting in forensic medicine. Applications: murder. Blood on a weapon of offense can be matched against the blood of victim. Similarly, blood stain on clothing or body of accused can be matched with that of victim. Hair roots found on a weapon can be matched against the blood of the victim and accused. Sexual offenses: seminal DNA obtained from vaginal aspirates or skin and clothing is compared with the blood specimen obtained from the accused. Paternity dispute: blood of child and alleged parents is matched. A child inherits 50% of DNA from mother and 50% from father. Auto rods of child DNA are first matched with the mother and then with the father, and if they correspond, then parents are biological mother and father of the child. Identification of the skeletal remains. In a case of mass disaster, accident, bomb blast, burned body, putrefied body, DNA from such bodies, if compared with the previous print, if available, or with the nearest relative like parent or children. Criminal cases: DNA obtained from other biological material like saliva, hair matched with accused. Confirmation of identity of exhumed dead body. Tracing pedigree and establishing family relationship. Biological identification: exonerate a falsely implication person of any crime. It has limitations also. If hybridization is improper or if activation of X-ray is less, band pattern of DNA may change. Inability to visualize patterns may lead to inaccurate interpretation. DNA profiles are very minute and their resolution may be very faint, and hence for judgment opinion of experts should be trusted. Absence of proper international guidelines making chances of errors are common. Very expensive and time consuming, cannot differentiate between identical twins. Medically legal importance. DNA fingerprinting is very commonly employed method in criminal cases to establish identity due to its specificity and sensitivity. Thank you.